So, the disadvantages of natural plant extraction, it includes the low concentration of the product in the plant. There may be, because of the demand supply difference, there can be limited supply. It can be an endangered plant because of extensive uprooting by the industry. Then it can be an endemic plant, not able to grow elsewhere, only in a particular region. So, there can be limited supply for these reasons. Now, endangered species, I said, can be a limitation which may lead to ultimately the extension of the plant. Then natural plant, because it is seasonal, the compound is temporal and spatial variation is there. So, because if it is seasonal, then there is long time required, maybe it only comes up when the mature plant comes up. So, non-uniform and inconsistent quality and quantity. So, what are the alternative production methods for such high value phytochemicals? Either some, as I said, we had seen how complex are these structures and the biosynthetic pathways. So, these with very high value and low volume metabolites, very complex structures, too many chiral centers. So, it has been observed that chemical synthesis is very difficult, and even if it is, people are able to do it, it becomes a multi-step process. So, biotechnology based methods, what can be biotech based methods? One is plant cell based bioprocess. What else? Which is not mentioned here. Protein engineering? Where? Means heterologous expression you are talking about. So, in some other host system, eukaryotic host system. But some of these secondary metabolites people are trying. The most frequently used system is yeast, where people, being a eukaryotic organism, people are trying to express. But the challenge is that some of these high value metabolites, the biosynthetic pathway is not elucidated. So, now how to get those intermediate steps? The protein engineering which is required, you need to know which proteins are involved. Then the other is, it is such a wide and big spectrum, like for example 40 or 50 steps are involved, and if it is a heterologous system where the pathway is not present, so all those steps have to be included. So, it is very challenging, which may cause the yields are very less, which can make the process non-economical and challenging. So, the most feasible process which is proposed is, which we would like to propose is plant cell based bioprocess, where the machinery is present, but you need to work around the different other bottlenecks, which can be the low yields and scale up of the process. So, the advantage of producing these high value metabolites which are low volume in nature using plant cell technology can be, there can be control of supply. Why? Because the product is independent of the availability of the plant itself, and it is away from climatic, geographical and governmental restrictions. Then high growth and productivity. So, high growth and productivity as compared to the natural plant. How? High growth and productivity as compared to natural plants.
Productivity, because we can culture them in less amount of time.
Right.
We can optimize the production according to our needs.
So, growth rate, because these are amenable to optimizations. So, you can end up, the time is the big factor. You can run batch to batch, it is months or years where you will have to wait. Here you can run a reactor and harvest. So, reduction in time and space is logical requirement for the production of your desired compounds. Now, strain improvement, it is also the cell line itself is amenable to optimization. So, you can manipulate the cellular yields to improve the YP by X, which is product yield per unit biomass. So, there is ease of extraction and purification by because you are doing process optimization. So, you will be optimizing the conditions so as to increase the productivity of your desired compounds. So, what happens that the relative abundance of your desired compound becomes more than the other small molecule or other impurities. So, thereby the downstream processing can become easier. So, what do we need for plant cell cultivation? We have now studied all this. An appropriate explant is needed, a suitable growth medium. What does the growth medium composed of? You will have major salts, minor salts, energy sources, growth regulators in it, which will fulfill the plant cell growth. And this can be what form? Liquid or semi-solids. So, aseptic conditions are required so as to prevent contamination, especially from microbes. Now, optimum culture conditions, so there will be external culture conditions, means environmental conditions, and other physical and chemical parameters. So, this is a schematic. How do we begin? We begin from the plant parts, then we bring it in the form of a dedifferentiated form, which is callus. From that callus, you can select the cell lines, bring it in suspension, do the optimizations where yield enhancement strategies are implemented so as to improve the YP/X. What kind of strategies come in this? Your improvement, your media optimization, precursor feeding, elicitation, and genetic engineering, which can be metabolic engineering. Then come to the bioreactor stage. Once the suspension has been optimized, at bioreactor stage what can be done? You again optimize the bioreactor operating parameters, which we also called as fermentation parameters. Then you can optimize the operation mode of cultivation, which means can be feeding strategies in order to remove what? Why do we have to have feeding strategies? What is the aim? Why are we talking about modes of cultivation here every time in the reactors? Why all the reactors cannot be run as batch? To remove nutrient limitation and nutrient inhibition if it is there. So, because in order to improve the productivity or yield you would like to increase the biomass or the product, and why in a batch it is getting limited? Because of the nutrient limitation. So, nutrient feeding is done to get over this nutrient limitation. And now with the logic that if nutrient limitation, you can dump in all the substrate so that it is never limited, so then inhibition can play a role. So, there comes the role of how do you feed, when do you feed. So, then after you have grown the biomass, it comes to downstream processing. Downstream processing means separation, purification of the product. So, what are the approaches which we are going to discuss which can lead to improvement in yield and productivity of the secondary metabolite? Optimization of media composition, optimization of environmental factors, addition of precursors, cell permeabilization, addition of elicitors, immobilization of plant cells, and high density cultivation in reactors. And in some of these is in order to reduce the hit and trial and minimize the number of experiments which you would like to conduct to reach to the optima, we will see how mathematical modeling is used with case studies. Now, yesterday if you remember, I was talking about that in order to start the bioprocess development, you would like to begin with the highest yielding cell line. So, logically you would like to pick up first the plant variety which is highest yielding. How is screening and selection? Is it same or is it different? Be it microbial, be it animal, be it plant cell fermentation, these techniques remain the same of optimization. When we say screening and selection, she was close. Screening is a passive technique, selection is an active technique. Screening is what she said for selection, in which you screen the cell line based on, how would you screen suppose? Any example? Okay, so you will engineer the cells first for blue white.
There are certain bacteria which can utilize the blood agar media and some which can't. So in this screening process, when we subject it to the same media, we can understand which are the cells which utilize the media and which are the ones which can't. So that's a screening process.
No, so extrapolate it to secondary metabolites from plant cells. We need to select the highest yielding callus line or the cell line? Callus is a mix of a number of cells, isn't it? That's why we say that callus would lead to difference in with subsequent subcultures. You may find that the yield keeps on varying. Bacteria, where is the bacteria coming?
Like say if the plant is producing a particular secondary metabolite for defense against the bacteria, that will be kind of a test organism to find out if the production is actually happening or not. You can use the bacteria to find.
On paper, this can be done. Think in terms of you are given and you need to develop a bioprocess. So think simple, which can cost you less, which can easily be implemented, and then you need to take the cell line. So once you start growing bacteria along with plant cells will not survive.
Ma'am, taking her point, we can just introduce some amount of stress conditions in which the secondary metabolite is produced, and we subject it to all of those callus lines, and we see which of them produces the most amount of secondary metabolites.
That's correct, but the only check here is that you have opted for a material which is known to produce that compound. You have a number of cell lines. So one is you check the yield of the product and you select the highest yielding cell line. And the other is, if suppose it is some of the if it is pigment based secondary metabolite, so then you can take that piece of callus which demonstrates that color. If it is a colored pigment, anthocyanins or any colored other pigment, then you know from that mixture of cells which cells will be producing that callus. Or as you people said, that if it is now coming on to selection, which is you would like to only take the desired cell lines, the others you do not care. So let them die, only the ones which you want to select should survive. It will be useful when you are selecting a plant cell line for what? Give me some examples, for what purpose? If I am trying to find a cell line which is highly resistant, suppose I am looking for salinity stress, as you were saying. So only those which can resist will survive, the others will not survive. So with subsequent subculturing and acclimatization also, you will end up having cell line which is resistant to the stress, and therefore you can pick up the desired trait cell line in the cell line. So now plant cell cultures, we know that they are genetically heterogeneous, means what? That the change in chromosomal, so that was what, when I say change in chromosomal number, arrangement, what kind of variation am I talking about? Somaclonal variation. So epigenetic instability means change in the gene expression or cellular phenotype caused by mechanisms other than changes in their DNA sequence. So which causes genetic instability leading to product accumulation only in some population of cells. That is the problem which the heterogeneous callus would have. Now productivity in such mixed population of cells is liable to be unstable. So generally when we induce from a explant a callus, it is not called as a plant cell line, because from the wound which you create and the cells start around that wound, the cells will start rapidly dividing and forming a de-differentiated mass. Now all these cells may have a different makeup, and as you would grow it on a solidified medium, they would multiply. So some of the cells which are close to the medium will have more approach to the nutrients, and the nutrients are more available to those cells than the cells which are at the top. So there are biochemical gradients which may form, which may lead to ultimately change in their biosynthetic potential. So it is generally observed that when you begin with callus lines, either you first bring it to suspension, and through the cell sieving, if you remember, I talked about the cell sieving technique, in order to get to a synchronous culture based on the size, with an assumption that the cells of same size and shape can be assumed to be in the same metabolic state. so thereby leading to a synchronous culture, or you keep subculturing them such that under the conditions which are preferable for the secondary metabolite of interest, such that during the subculture natural selection happens. So, you will only the cells which are capable of producing under those conditions the secondary metabolite would continue to grow, and later they would be the majority. So, it is always the kind of cells in the callus and the number of those cells in that callus which are capable of producing your secondary metabolite. So, how to increase that number? And it is generally observed that when you begin with the callus induction, the yields for initial a year or so, you will see a large variation. Only after 3-4 years of subculture, you will find that it comes down to a consistent product yield. So, productivity in such mixed population of cells is liable to be unstable, since change in any factor, that is the very reason why we in lab always insist on keeping the conditions consistent. You cannot take lightly the subculturing of the callus, the parent material, which is going to form your master cell bank. So, under the conditions which you subculture, the time at which you subculture, you cannot think that if it is 30 days, let me do it in 40 days, it does not matter. Everything matters, the time at which you are subculturing, the conditions. Today you had put 0.8, tomorrow you had put 0.6, it is going to vary. So, one has to be really careful while subculturing your master cell bank. Since changes in any factor, environmental or nutritional, which favors the growth of a particular subpopulation of cells, thereby changing the composition of the culture. The variation in culture, now because it is something which can tweak the biosynthetic potential of your cell mass, so it also becomes a tool for variation. So, the variation in the culture offers the opportunity of isolating from the mixed population of variant cells the ones with high productive capacity, and thus developing from them cell lines of high productivity. So, the rate of accumulation within the productive cells and the proportion of such cells in the culture is what which governs the yield or selection of the high yielding cell line. The low production in cell culture can be due to, one is competition between the primary and the secondary pathways. The same intermediates as we have seen in the pathways that then bifurcate for secondary metabolism. So, there will always be a competition of carbon flux. Lower expression of key enzymes in the rate limiting steps in the pathway or gene expression of the secondary metabolism can be a limitation. Then screening is a passive technique by which a number of cells are analyzed for the desired trait. So, we generally, as you were saying, we would look for the highest yielding cell line, we will do some part of the callus we will take out, we will do extraction and we will do yield in analysis. Or if it is a colored pigment, you can directly view it from your, it is visible to eyes. If it fluoresces under UV, then too you can do it like UV light. So, whether the callus which you have obtained is producing your compound or not, you can check. Now, selection is what? Selection is an active process by which it deliberately favors only the survival of the desired variant, and the wild other type of cells, they will die. So, callus systems, they are used for screening and selection. Cell lines which accumulate high levels of colored compounds can be selected through visual observation. The callus can be checked visually for altered pigmentation by the naked eye for colored spots under UV lamp. Screening of pigments, for example, if suppose you know from literature that greener is the callus, it is somewhere linked to secondary metabolism of secondary metabolite biosynthesis. So, you would like to select the most green callus line. So, screening on the basis of pigments like chlorophyll, anthocyanins, naphthoquinones, carotenoids, these are all colored pigments. So, if you are looking for such kind of pigments, it becomes very easy to do the screening. Now selection can also be performed by using your screening techniques. Screening means using different mesh sizes and trying to reduce the mesh size, for example, from some 100 microns to less than 100. So, suppose 500, greater than 500 microns to less than 100 microns. So, the same thing that you can end up in a synchronous culture. The selection can be performed with a fine rapidly growing suspension consisting of aggregates of 50 cells or so. Now, fine cell suspension is suitable in selection for resistance. Why do you think every time the stress is on fine cell suspension for selection processes? How will it help than the lump culture? We are talking in terms of selection. Selection means?
So the size variation will not affect the selection process. Like all the cells will have the same.
That was cell sieving. Now in general there can be more than apart from cell sieving methods directly, like for example if you are looking for resistance, so the rest will get killed, only those which are resistant will survive. Either this resistance can be salt resistance or can be against the toxic metabolite or anti-metabolite resistance. So if suppose the other get killed and you need to, now the only one survives, why it is recommended to use dilute cell suspensions? Like you were giving an example for the bacteria, you grow it in a solidified medium such that only the bacteria which can degrade that particular metabolite or which is resistant would live. And then what do? You pick up what? You pick up the colony. So there also what is recommended? To use dilute solutions. You can get to, the purpose is what? Selecting the highest yielding cell line. A callus is what? A heterogeneous mass of cells. So therefore what is preferred? You bring it in suspension, make it dilute, then select for a resistant cell line and allow that to grow and multiply to give you a fresh callus using what? Single cell culture techniques which we have learnt before. So in the selection program for resistance in cell culture, one can detect growth of the resistant population, but because you end up in single cells, it will take time for the callus to reappear and for the culture to again come back to a rapidly growing state. So therefore it is said that it will take some 1 or 2 months to come back to reach to a uniform callus, a single cell based callus line. Now to avoid inherent problems in the use of callus or cell culture for selection and screening, the plating of cells or protoplast on solid media or matrices can be preferred. Now in plating the cells, the first step is to prepare single cells or small size aggregates for plating. The cells have to be plated at lower densities, allowing growth of individual clearly separated colonies that can be isolated and then subcultured. Unlike microbial cells, cultured plant cells require minimum inoculum density. This also we studied, that plant cells will be needing minimum inoculum density for the growth to begin. Generally plant cells are grow, they communicate with each other through
Plasmodesmata.
So there is cell to cell contact which is necessary, there is communication happening. So to facilitate and bring it to that condition, that minimum inoculum density is needed. Now preparation of single cells or protoplasts we have already studied. To filter successively cells of fine suspension culture through, so we use cell sieving techniques. So where around 500 to 75 microns you keep passing the cell suspension. This will end up in synchronous culture. The obtained suspension will consist of single cells or small aggregates which most likely have originated from single cells. Why? How can you assume that? When we are using these cell sieving techniques, we are starting from big size and we are ending up at a very small size. 75 microns is a less size. So, generally the cells, we said 50 or 100 cells together would be there in cell aggregates, and the size is around plant cell size. That also we did. Average. If you know that average, you will get to the logic why this sieving technique of 500 to 75 will end up in either single cells or will end up in very small aggregates, which would obviously be coming from the single cells because they have multiplied and form a little bigger than the single cell. So, large aggregates suspension adding pectinase helps. How? This also we have done. That is the very reason we were doing all that. Generally, you will find that even when you will induce a callus from different types of explant, they will all vary not only in their biochemical makeup, they will also sometimes vary in their morphogenetic or morphology. Some will be very hard and compact, some will be very friable that you will just touch with a spatula and they will disperse in the medium. So, that is because of the friability, is because of the water content in the callus. So, large aggregate suspensions we are using pectinase now because it is preferred that the callus should be friable in nature if you want to generate a suspension, because it can easily disperse in the liquid medium. Screening and selection should be done from a freshly prepared cell suspension from the plant than from a very old suspension. Why? Why should you prefer young? Because they will be more actively growing cells. The most ideal tissue for isolation of single cell is leaf and mesophyll cells, which can be mechanically and enzymatically isolated. In order to select a single cell for plating, cell suspensions must be diluted to densities where the plant cells do not grow without special supplementation. So, what are the methods of screening and selection? Secondary metabolites, it may show an intense color. So, visual detection, you can take that piece of the callus and start propagating that callus. So, you will end up in a callus line which is giving the product of interest. Altered pigmented areas of the callus can be easily detected, or direct analysis of the cell extract is done to know the quality of the clone. The clones under investigations have to be divided for subculture and chemical analysis.